Hi, everybody. Thank you for joining us. Today, it's with great pleasure that I introduce today's seminar speaker. Dr. Jess Cardin is an associate professor of the Department of Neuroscience at the Yale University School of Medicine, and she's also a member of the Kavli Institute for Neuroscience at Yale. She is the recipient of many distinguished awards, and I'm not going to waste time listing it all, but just for some examples, she has the McKnight Scholar Award, the Smith Family Award for Excellence in Biomedical Research, and the Alfred P. Sloan Foundation Fellowship. She's also on the Scientific Advisory Board at the Allen Institute for Brain Science. And I don't want to take up any more of her time, so I would just say that Jess is always performing cutting edge research. And so it's not surprising that she has already contributed to some of our understanding of the neurobiological basis of cortical function. She specifically contributed to our understanding of the roles of the diverse cell types that we have and the cortical motifs in the brain. And I think we will hear some of that work today. Her title is State Dependence and Neuromodulatory Signaling in the Cortex. And thank you, Jess, and welcome.
Thank you so much for the kind introduction, Chayu, and for the invitation to come and share some science with all of you. And I am very sorry that I don't have an opportunity to meet you all in person. But instead, I will just encourage you to please interrupt me if you have questions. I can talk all day, so it's much more fun when you ask questions as they come up, or you can save them till the end. Or, if the students are more comfortable putting questions in the chat, please go with that, and we'll stop periodically and answer any questions that came up. So as Chayu said, we think a lot about cortex and cortical circuits. And what I'm going to do in this talk today is, instead of telling you just one story, I'm going to tell you about some ideas and some stories that span across scales. And so I'm going to start by talking about local circuits, small circuits in one part of the cortex. And then in the second half of the talk, I'm going to try and take a step back and talk about how some of the things that we see in local circuits may or may not be consistent across the whole cortex when we look at multiple areas. So one of the things that we think about a lot in my lab is the idea of flexibility. And if you think about all the things that you do on a daily basis, all the experiences you have, all the places you've been in your life, all the things you had to learn, if you had a dedicated circuit in your brain that was specific, specialized for that thing, right? You had one of those for every experience, every learning event, every environment that you encountered. You would be dragging around a brain the size of a truck, right? So instead of having a brain full of specialist circuits, you instead have a brain full of general circuits, circuits that exhibit a huge amount of flexibility in their function and are able to perform different operations, different types of information processing computations at different moments in response to changes in environmental input or internal brain state, neuromodulatory state, cognitive and behavioral demands. And we're really interested in understanding what elements of a circuit can be flexible, what promotes flexibility in local circuits, how that extends to large scale circuits across the brain, and what that means for behavior and for cognition. So what we're hoping to understand is, as you got out of bed this morning and hopefully you brushed your teeth and you had your first cup of coffee and you woke up, but then you stepped out the door of your house and you went to work, and maybe you had a second cup of coffee if you're like me. all of the ways in which these generalist circuits in your brain change their activity with changes in your internal behavioral state and with your external interactions, behavioral interactions with the world around you. Now, we cannot do this invasively in humans because we are interested in all those different cell types, and we really would like to get into circuits and manipulate them. So instead, our model organism of choice is the mouse. And work in mouse cortex over the last decade or so has really focused on some core motifs, core components of local circuits. And I'll talk about a few of these in the first half of this talk. But in any cortical circuit, we know that there are excitatory pyramidal neurons shown here in green, as well as multiple types of inhibitory GABAergic interneurons shown here in blue. I'm only highlighting these supergroups here. I'm only highlighting the broad general groups. Each of these groups of neurons includes many subgroups or subtypes of neurons, especially for the interneurons. But here we have excitatory neurons in green and three main flavors of inhibitory interneurons in blue: the soma targeting parvalbumin positive fast spiking interneurons. These are the classic well-studied interneurons. These are the ones that you can record beautifully with extracellular electrodes, and we have known about them and recorded them for many, many decades now. And also we have the harder to find dendrite targeting interneurons, the somatostatin positive cells, and the VIP vasoactive intestinal peptide expressing interneurons. And these predominantly target the dendrites of excitatory neurons, but you'll notice here that all these interneurons interact with each other as well. So this is actually a very complicated microcircuit in the brain. And this motif of excitatory inhibitory connectivity is fairly standard, fairly well replicated across many cortical areas. To get into this circuit in the brain in active manner, we need to capture the brain in the act of being flexible, right? And the way that we do that is by taking advantage of the naturalistic behavior of mice. So as you can see in this video, this is a head-fixed mouse sitting on a wheel. So the mouse can sit or run as it chooses. It's free to exhibit natural behaviors. And mice go through regular cycles. They sit, they kind of fall asleep. They sort of become more aroused and active and start running. And they will do this over and over and over again many times within one experiment. And if you watch this video, you'll see that we have many external markers of behavioral state that include running, which is an obvious one, but also facial motion. You'll see that the face will move even if the mouse isn't running. Sometimes if the mouse becomes moderately aroused, you'll see the face will start to move even if the mouse hasn't started running yet. And we also use the pupil, which, as you can see at the moment, when the mouse is very aroused and active, the pupil is very dilated. If I back this up and show you the beginning of the video again, you see when the mouse is very quiescent here at the beginning, pupil is very constricted. We generally tend to think about this state, this very deeply quiescent state, as being very similar to an undergraduate student in the back of a large lecture hall. Really, really bored, kind of zoned out, not paying a lot of attention, not processing a lot of sensory information from the environment. Whereas the more aroused the mouse gets, the more interaction it has with the environment and the more sensory information it's taking in, and possibly the more accurate the brain is in representing that information. Now, if we look more closely at what happens at these transitions, this is data from an experiment in which we have video of the eye. We also have a measurement of how much the wheel is moving, so how much running the mouse is doing. This very large trace here in the middle is the local field potential, so a measure of population synaptic activity, a synchronous population synaptic activity. And at the bottom is the raster of one identified putative excitatory neuron in the cortex. And so when this green line is flat, the mouse is sitting very still. And you'll see very big, slow events in the local field potential, moderate level of firing of the cortical neurons. And there's going to be a moment here where this green line that denotes the wheel is going to start wiggling. And this is just before the mouse starts to run. And you'll see just at this point where the mouse becomes a bit more aroused, the pupil dilates, the local field potential desynchronizes and goes from big, low frequency, slow events to small amplitude, high frequency events. And the firing rate of that neuron increases tremendously. Right? And then the mouse gets a little bored or stops running. Right? It sits back down again, and you'll see the firing rate will go down. The local field potential will pick up those big, slow events again. So the frequencies will get lower in the local field potential here, and the pupil constricts. Right? So data like these, shown again in a more fixed form here, from the pupil dilation, from the running on the wheel, the single unit firing, as well as the local field potential activity here on the bottom. All this data so strongly suggests that activity in the cortex is very robustly modulated by behavioral state. So when the behavioral state changes, activity in the cortex changes too. Now, population.
Sorry, Jess.
Go for it.
Pablo Fuente Alba has a question. Where's the local field potential recorded? Which area?
So all of these recordings are in primary visual cortex. And this is a relatively superficial depth bipolar from superficial cortex, probably layer 3 in this case. Most of our local field potentials are depth bipolars compared to the surface of the cortex. And all these recordings that I'm showing you here as population averages are also from primary visual cortex, but you can see this kind of relationship in many cortical areas. So as the mouse gets ready to start running, you see there's an anticipatory increase in firing rate shown here in black. So population firing rate is a one neuron shown in black here, population firing rate of the excitatory neurons in blue here, coming up to the onset of locomotion and then stays up while the mouse is running. Now in orange, we have our putative fast spiking, probably parvalbumin positive interneurons. This is extracellular recording, so we don't have genetic tagging for these interneurons. We're dividing them up by their action potential waveforms. So fast spiking cells statistically are most likely to be parvalbumin positive basket cells. But these also have an anticipatory increase in firing rate, and they stay modestly elevated as the mouse starts to run. And you can see at the same time, the wheel speed, the mouse is beginning to run pretty consistently, and the pupil is dilating in the same anticipatory way. So arousal precedes locomotion here pretty robustly, and firing rate tracks with arousal, not just with motor activity in running. But I said at the beginning, right, that there are multiple different types of, especially GABAergic interneurons in these circuits. And this population of parvalbumin positive cells are the only ones we can really reliably identify with extracellular recordings. These other two groups of inhibitory interneurons are very hard to record from. We could do it, but it would take a very long time because they, especially the VIP cells, are very sparsely represented in the cortex. They represent about 1 to 2% of all cortical neurons. So they're in there as if you sprinkled raisins in your pudding. And they are very statistically unlikely to be recorded. So instead of recording them with electrophysiology, we turned to genetically targeted calcium imaging in vivo. And so here we are expressing the calcium indicator GCAMP specifically in the VIP interneurons as an example. This is targeted using a Cre-dependent viral vector carrying GCAMP and a VIP Cre mouse line. And the mouse is sitting in exactly the same kind of setup. It's head fixed, it's on a wheel, same wheel. But in this case, instead of electrodes, the mouse is sitting under a 2-photon objective. And here you can see in this video in green are the VIP cells. So GCAMP fluorescence is a proxy, a stand-in for the firing activity of the neurons. So when these neurons are more active, we see them fluoresce in the green. When they're less active, you see them fade away into the background. The trace on the bottom is the running speed of the mouse. And you can see in this video that when the mouse is not running, all of these neurons, the whole population of VIP cells fades into the background. When the mouse starts to run, the whole population of interneurons becomes simultaneously active, suggesting that these interneurons are also robustly modified. Their activity is modulated by changes in behavioral state.
Sorry, Jess? Do you know if these also show that ramping up before locomotion?
They show somewhat less ramping up, the VIP cells do, which is kind of interesting because we tend to, I'll get to some neuromodulatory stuff in the second half of this talk, and we really think that the ramping up is partly driven by increases in neuromodulators like acetylcholine, and these cells are very sensitive to acetylcholine, and yet we really don't see them doing this ramping, but the other population of dendrite targeting interneurons, the somatostatin cells, has more of a ramping effect. So actually right now we're doing dual population imaging experiments with two at a time populations of interneurons, trying to look in simultaneous imaging experiments at the relative timing of activity. It's very hard to compare across populations from non-simultaneous data. Okay, this isn't crucial here. This is just a quantification of the type of data I just showed you in that video. So the important plot is the one here in the middle, where blue indicates no modulation or negative modulation, and red indicates positive modulation of the activity of the VIP interneurons. If you see this white dotted line here indicates the onset of locomotion, and you can see that basically the entire population of VIP cells comes online when the mouse becomes active and starts to run. And this is very consistent across populations and across animals. Okay, so all of that data is very beautiful. And what it tells us is that the activity of many, probably most of the populations in the cortex is modulated by changes in behavioral state. So all of these neurons are involved in the functional flexibility of the circuit. But that doesn't tell us whether the circuit is using incoming information differently, performing different computations under those different circumstances. All it tells us is that firing activity of neurons has changed. So to answer that question, we can change the experiment a little bit and show some stimuli. And so here, because we're in visual cortex, we are showing visual stimuli. And we're looking at the relationship between the signal, how much visual response there is in the neuron's activity compared to its background activity or noise. And we've divided this into excitatory cells in blue and inhibitory cells in orange. So here A is aroused or active. So this is during running. And Q is quiescent, so sitting still and very, very quiet. And the value on the y-axis is the signal to noise ratio for the cells. And the bottom line here is that this measure and many other versions of this that we've done over the years suggest that cortical neurons do a better job of encoding stimulus information, sensory stimulus information, when the mouse is more aroused. So this is also true if you look at noise correlations. So noise correlations are a form of shared synchronous activity pairwise across neurons in a population, and they're thought to be detrimental to the encoding of sensory stimuli. And in fact, noise correlations, as shown here, are much higher during the quiescent state than during the aroused state, again, suggesting that sensory encoding is improved with arousal. Okay, so neurons are indeed performing different computations in different behavioral states. However, what we really want to know is the mouse using that information differently in different behavioral states? Does this change in neural activity and change in neural computation, the change in encoding of sensory stimuli, mean that there is a change in the behavioral outcome or the way the animal is acting on that information? So what we've done in recent years is to use a visual detection task in mice sitting in the same head fixed preparation. So these mice are sitting on the wheel and looking at a stimulus on a screen and performing a go, no go type of task. The go stimulus is a drifting grating on the screen, and the no go stimulus is a blank screen. And here the go stimuli vary in intensity or contrast on the screen. So they range from very, very faint, very hard to detect, to very, very strong, very easy to detect. And when the animal gets it right, licks in response to a stimulus, it gets a drop of water. So all of these mice are water deprived before training on the task. Okay. So this is an example of what a trial on this task looks like. So this is mouse head fixed on the wheel. in the middle of a session performing this task. It's waiting for the stimulus to arrive on the screen. It gets an auditory cue. You can just barely see that drifting grating there on the screen, and the mouse licked correctly and got a drop of water. And mice will do a few hundred trials of this task in a session. One of the things that we noticed about the data from these experiments was that, in fact, there was a lot of diversity, a big spread in the behavioral states that we were seeing. So when we looked at pupil diameter, for instance, on the x-axis here, and we looked at how that correlated with the animal's running behavior, what we found was that there were, in blue, periods when the mouse was sitting still. And we divided these into low arousal sitting periods and high arousal sitting periods. So 2 different shades of blue here. So there are very, very quiescent states where the animal is sitting and the pupil is very constricted. And there are moments of moderate arousal in the lighter blue, where the mouse is sitting still, but the pupil is dilated a bit, and the mouse shows evidence of being moderately aroused. And then in orange, we also have periods where the mouse is highly aroused. On average, the pupil is more dilated, and the mouse also runs. So we've divided our behavioral data into those 3 states, and this task gives us 4 different types of trials. Correct hits, where the mouse saw a stimulus and licked for a water reward. Incorrect misses, where the mouse sat there the whole time and completely missed seeing the visual stimulus on the screen and never licked for a reward. Correct rejections, where the mouse did not lick to a no-go stimulus of a blank gray screen. And false alarms, where the mouse licked even though no stimulus was presented. And here in this plot, what I'm showing you is the probability of stimulus detection on the y-axis and the intensity or contrast of the stimulus on the x-axis. And this is again divided into those 3 levels of arousal that we see, from very, very deeply quiescent in dark blue to moderately aroused in light blue to very aroused and running in orange. And this is the performance of a population of mice on the task. And what you can see here is that the performance of the mice gets better with more arousal. The whole psychophysical curve shifts to the left as the mouse becomes more aroused. And the threshold for detecting the low contrast visual stimuli actually gets lower. So the mice do a better job of detecting low intensity stimuli the more aroused they get. And the quantification for that is here on the right-hand side. So this is the C50, or the 50% detection point for visual contrast. And you can see that it goes down significantly, in fact, with arousal. So activity in the cortex of both excitatory and inhibitory neurons is changed with behavioral state. This is correlated with improvements with arousal in the encoding of sensory stimuli, and also with improved psychophysical perceptual performance on a visual detection task as the animal becomes more aroused. Okay. So this suggests that the state-dependent modification of the activity and the neural computations within this small circuit in the cortex really have a profound impact on the way that information is being used during perception and behavior by the animal. And of course, as I said before, this circuit, in its general form, exists all across the cortex, suggesting that these state-dependent modifications of information processing and behavior may be a common theme in many cortical circuits. So here we find that circuit function varies with waking behavioral states, such as arousal and locomotion. We know that both GABAergic and glutamatergic neurons are modulated strongly by behavioral state change. That includes the firing rates and the visual responses, of course. And this modulation of cortical activity is connected to modulation of visual perceptual performance. Okay, so this is really beautiful data from one particular cortical area, right? So all of the methods and approaches that I just showed you are very good at identifying these motifs of activity on a small scale at the cellular level in primary visual cortex, for instance. But they do not allow us to answer questions about whether these state-dependent changes in functional flexibility are the same everywhere in the cortex, or if these things are unique to, for instance, primary sensory cortex. To answer those questions about the broader common mechanisms of state-dependent change across the whole cortex, we really need a completely different set of methods. So we can't do it with these methods at all. And as I mentioned sort of in passing in some of the earlier slides, when we think about state-dependent modulation, one of the global mechanisms that quite obviously comes to mind is neuromodulation. So one of the mechanisms that has been long identified in playing a big role in state-dependent modulation is neuromodulators like acetylcholine and norepinephrine. But there are multiple possibilities here. So one is that neuromodulatory signals communicate with the entire cortex. All of those little microcircuits all over the cortex might see the same neuromodulatory signal. Like, aha, now we're going to wake up. And everybody wakes up exactly the same way. Alternatively, it might be that those neuromodulatory signals are not the same everywhere, that they are spatially or temporally dynamic in different ways across the cortex. And that would suggest that different parts of the cortical circuit, the large scale circuit in the cortex, might be differently modulated by changes in behavioral state. We'd really like to better understand the influence of neuromodulatory signals in particular on behavioral state dependent changes in cortical activity. So we'd like to be able to measure the spatiotemporal dynamics of acetylcholine, for instance, across the whole cortex, as opposed to in one cortical area. And we'd like to better understand the relationship between neuromodulatory inputs and these changes in cortical activity that we have identified at the local level. Okay. So here I'm going to take a very brief detour. So this is a good moment for questions. If anyone has lingering questions about the first part of the talk, I'm going to take a 2-slide detour and talk about some methods for just a moment. But if you have questions, please stop me. So the calcium imaging that I showed you a few slides ago, it should be pretty familiar as a very standard technique in neuroscience these days. And we often use either the green calcium indicator GCAMP, which is the product of a molecule of GFP coupled to calmodulin, calcium sensor, right, binds calcium and causes a change in fluorescence. Or in the red, we use the red indicator RCAMP or sometimes RGECO, which are mRuby or mApple, also coupled to calmodulin as a calcium sensor. Okay. We need to sense not only neural activity, which we can do using these calcium indicators, but also neuromodulatory signaling. To sense neuromodulatory signaling, there's a whole slate of new tools that have recently been developed. And the one I'm going to talk about the most here is a tool called Cholinergic grabs tool. So ACH 3.0 is a molecule of GFP coupled to a muscarinic acetylcholine receptor, which has no biochemical activity. So it is membrane inserted. It robustly binds acetylcholine and fluoresces upon binding, but does not have any biochemical activity within the cell. So if you express GCAMP in a neuron, right, GCAMP is floating around inside the neuron. And let's say the neuron fires some action potentials. Intracellular calcium signals rise, right? So calcium binds to GCAMP and causes a change in fluorescence. So you see an increase in fluorescence in the green. Right? This is quite different from the way that the ACH 3.0, the cholinergic grabs tool, works. In this case, we have a membrane-bound protein, so it is sensing the extracellular space, not the intracellular space. It is binding acetylcholine because it's a modified muscarinic receptor sitting on the membrane, and binds acetylcholine and fluoresces in the green. So you see a conformational change and you see increased green fluorescence when acetylcholine is bound. But the key difference here is that we're not sensing intracellular activity. We are instead hijacking neurons across the brain and turning them into sensors of extracellular acetylcholine release. So this allows us to measure cholinergic release in the cortex in a very broad scale, which I'm going to show you in the next couple of slides, by putting this sensor in the membrane of lots and lots of neurons. Now, the way that we have done this in recent work is by using a set of techniques that was developed as part of a large collaborative effort here at Yale, using early postnatal injection of AAV virus. So this is injection of high volumes of AAV9 virus into the transverse sinuses of P0 to P1 mouse pups, which results in whole brain, very robust expression of whatever viral construct was injected. So the example up here is shown for GCamp, but on bottom, I'm showing you an example of brain in which we co-expressed throughout the brain. In the green, the cholinergic indicator ACH 3.0. So this is the indicator for acetylcholine across the cortex. And in the same brain, we have the red calcium indicator RCamp. So in this preparation, we can simultaneously image neural activity locally or on the broad scale in red and acetylcholine release in green. For these experiments, we're taking advantage of a different imaging technique. So this is a 1-photon imaging technique rather than 2-photon. The mouse is sitting under a couple of CMOS cameras and some LEDs in the same head fix preparation on the same running wheel as shown here. And we are flashing LED light on top of the entire cortex of the mouse. And this is the top-down view. So here we're able to see most of the dorsal cortical mantle across many cortical areas simultaneously. And in this GCamp imaging example, you can see patterns of neural activity across the entire cortex as the mouse performs all the same spontaneous behaviors. Okay, so in the data that I'm going to tell you about here, I just want to mention briefly before I start showing you data plots that what we see here is sort of the whole cortex picture. And in order to analyze it, most of the time, fairly standard in the field, people will parcellate this data. So sort of use a consensus atlas map generated by the Allen Institute, which is shown here, to divide up the image into cortical areas. So we're using the Common Coordinate Framework version 3, which has 30 odd main cortical parcels. There are other ways to do this, maybe even better ways to do this. So if anyone is really interested in the technical details of functional versus anatomical parcellation, I'm really happy to talk about that at the end of the talk. For the purposes of this data, we've just used the Allen Common Coordinate Framework, and I'm going to show you mostly examples from M1, primary motor cortex or secondary motor cortex, M1 or M2, and B1. Okay. So just to sort of show you some good evidence that what we're looking at when we look at the cholinergic grab signal, that ACH 3.0, is in fact cholinergic release. We did some basic control experiments where we put a bipolar stimulation electrode in the basal forebrain to stimulate cholinergic projection neurons throughout the cortex. And you see down here on the bottom that before we stimulate, we see very little signal across the cortex. However, just after we stimulate, we see a sort of homogeneous signal across the entire cortex as acetylcholine is released. We also find that when we stimulate the basal forebrain and look at the activity, not just of the grab signal in the cortex, but also our behavioral state indicators, we find that upon basal forebrain stimulation, we see an increase in cholinergic signal in the cortex. We also see dilation of the pupil, an increase in facial motion with whisking, and decorrelation of the EEG. So here we have an ECOG electrode just above posterior cortex on one hemisphere. So all signs point to this being a good marker for acetylcholine release. Okay. So this is what these experiments really look like. What we have is video of the face and the eye of the mouse. So we can see the pupil, we can see facial motion, which are both indicators of behavioral state. In one channel, we have ACH 3.0 signal. So this is cholinergic release across the cortex. In the other channel, we have RCamp, which is a calcium indicator giving us neural activity signals. And you can see in real time here that the cholinergic signal, the neural activity signal, are correlated sometimes and not correlated at other times. They vary according to behavioral state. So here you can see that there are fluctuations in facial motion and locomotion and pupil area. And those are sort of diversely related to the cholinergic and the calcium signals. So there's a lot going on here. But one of the things that you can see just from looking at the semi-raw data here is that cholinergic signaling is in fact not a monolithic signal across the entire cortex. When we see cholinergic signaling, it is not always lighting up, the entire cortical mantle. Instead, we see a high degree of spatiotemporal dynamics under different conditions. And so the data that I'm going to show you in the next couple of slides is unpacking when and where we see that spatiotemporal diversity of dynamics. Okay, so these are the three postdocs here who did this work: Sweta Lohani, Andrew Moberly, and Hadass Benisti. And this was really an enormous amount of heavy lifting by all three of them. So here are some example frame shots from the cholinergic signal on top and the calcium activity signal on the bottom. And then in red, so primary visual cortex, V1 will always be in red. M2, secondary motor cortex, will always be in purple. And here I'm showing you V1 and M2 and the correlation between them here for the cholinergic signal on the top and for the calcium activity next. And then I'm showing you all the behavioral state indicators: the pupil, the wheel speed, facial motion activity, for which we're using the FaceMap software that was developed by Carson Stringer. And on the bottom, frequency bands from the ECOG recording. So this is only to show you that this produces an incredibly rich type of data set. Okay, so from the earlier data that I showed you in the first part of the talk, as well as from these data, what we see consistently in the mice on the wheel is that they go through multiple cycles over and over again of essentially three types of behavioral state. We see a very quiescent state here with low arousal and very low metrics for those behavioral indicators of state. We see a state that is indicative of moderate levels of arousal where the mouse is not running, but for instance, the pupil is partially dilated and we see a lot of facial motion energy going on. And then we see a state of high arousal where the mouse has all of the markers of high arousal. The pupil is fully dilated, the face is moving, and the mouse is running on the wheel. And so we're going to compare these three distinct behavioral states in our data set. And of course, one of the first questions one might ask is, do these behavioral variables have any predictive value? Do they predict patterns of cholinergic release or patterns of neural activity? So we did a very straightforward regression analysis using full recording data sets and found, as shown here, that in fact, in large part, the behavioral variables do a terrible job of predicting the spatiotemporal profile of either acetylcholine or neural activity, but by far facial motion energy does the best. So facial motion energy predicts some of the cholinergic signaling and some of the calcium activity quite well, but not in the same part of the cortex, as you can see from these plots. So just using these behavioral variables as a way to understand the patterns of neuromodulation and neural activity is not going to be super productive. But remember, this is just a regression analysis where we threw everything all in at the same time. We can do better than that. Okay, now if you actually look at the data, which I highly recommend, by the way, looking at the data has almost always been the thing that allowed us to better understand what was going on. So if you look at the sort of raw behavioral data here, what you notice is that there are two types of events going on, right? We have very long periods of sustained behavioral state. So for instance, here we have one very long period of locomotion where the mouse is sitting, very, very quietly for a long time, and then it has a long locomotion back over here. Same for the face map, the high facial motion energy, we have low and high states. We also have transition points where the mouse makes a transition from one state to another. So we've separated out those different types of event in our data. And first I'm going to show you the transient changes in behavioral state. So this is just data aligned to transitions on the left-hand side here from quiescent sitting to high facial motion, so this modest level of arousal. On the top are cholinergic signals, and in the middle here are the ARCAM neural activity signals. So when the mouse goes from sitting quietly to whisking its whiskers or moving its face a little bit, we see a pretty big jump in acetylcholine and a little tiny bump in neural activity. And you can see here the mouse isn't running. You only see an increase in facial motion energy. Okay. On the other hand, when the mouse runs, we see a modest bump in cholinergic, peak cholinergic activity here, and a huge increase in neural activity. So this kind of clued us in a little bit looking at these transition points that there are some differences in the pattern of recruitment of neuromodulatory signaling versus neural activity in these different states. So now we're going to switch gears and look at the sustained periods of behavioral states. So these are moments where the animal spends a lot of time sitting quietly or with high facial motion or runs for a long period of time. And what we find is that the cholinergic signal shown here on the top row and the neural activity shown on the bottom row show different state-dependent dynamics across these three states. So I'm going to walk through each one. So let's take the cholinergic signal first. So when the mouse has low facial motion energy, so this on the left-hand side, low face map means the mouse is sitting still and it's not even moving its face. It's just zoned out like that undergraduate at the back of the large lecture hall. Right? And here, essentially nothing is going on, right? There's no cholinergic release really at all. Then we have the intermediate arousal state where the mouse has started to move its face. And here we have a very large increase in cholinergic release. And you can see that there is an especially large increase in the frontal cortical areas. Sorry, for those who don't do mouse work, I should have specified earlier, the nose would be at the top here. This is rostral, and the tail would be down here. This is caudal. Now, when the mouse starts to run, actually, we do not see a significant change, again, in the cholinergic signal between this intermediate and high level of arousal. And I see a hand up.
I'm sorry to interrupt you, but just I wanted to be clear to myself and be aware that I'm following you. So we have the GCOM in all pyramidal cells. Yeah, yours, because
right. Yeah, so so both of these tools are actually being expressed in all neurons, pretty much. They're both under, yeah, they're used under the synapsin promoters. So okay, the RCamp signal is like 80% excitatory neurons. For the cholinergic signal, it kind of doesn't matter what we express it in because we're sampling the extracellular space.
Mm-hmm. And the other question was can you explain me a little more about the face map, because I don't understand too well, and that also contains like the pupil dilatation or diameter.
That's a really good point. So if we go back here, can you see my cursor?
Yeah, I can see.
Yeah. Okay.
Thanks.
So from a video like this, we would extract two separate things. One would be a little chunk of image that just covers the eye and the pupil. Mm-hmm. And the other is a chunk of video that covers sort of the middle part of the face and part of the whisker pad, right? And so what we're noting here as face map is really an analysis of the facial motion energy. So we're taking the motion energy from the video over the middle of the mouse's face, calculating the motion energy. We're doing a little bit of dimensionality reduction here. So we're just taking the first couple principal components of the motion energy in the video. That means that signal probably includes both active whisking as the mouse is moving its whiskers, and also other facial movements. Right?
It does include like these ear things?
It does not include the ears, right? Yeah, but the ears are really interesting. We can pull the ears out with other approaches, including deep lab cut. The position of the ears is a whole other story. Okay. So here, when I'm showing a low face map, that means the mouse is sitting still and we don't even have any facial motion. Okay. Right? If we're showing high face map, that means the mouse is sitting still, but has lots of stuff going on with facial motion, right? It might be whisking a little bit, it might just be moving its face. Right. Now, locomotion is always paired with whisking. Mice always whisk when they run. So if you looked at the behavioral variables, you'd see that when locomotion is high, facial motion energy is also always high. Right? So that's how we've divided these behavioral states based on the fact that you can see nothing at all. You can see face alone, or you can see running, which also always has face and pupil dilation and everything else. Right? So basically low, medium, and high arousal states.
Okay, now I get it. Thanks.
Okay, good. And so interestingly for this, right, we see a big increase in cholinergic release with the transition from sitting to high facial motion, but no additional increase in cholinergic release when the mouse starts running. In contrast, if you look at this bottom row, when the mouse is sitting still versus when the facial motion energy is high, we see a little increase, right? A significant increase in calcium activity, which means the neurons are firing more. And then we see another significant increase when the mouse starts to run. So we see a bump in neural activity with every increase in arousal, but we only see a bump in cholinergic release in this first transition from low to medium arousal. Okay. So that just tells us about the amplitude of activity, right? It doesn't tell us anything about the relationship of signals across the cortex. So because we have all of the areas in the cortex recorded simultaneously in each of these channels, we can look at the correlations between areas. And so here I'm showing you as an example, the cholinergic signal in primary visual cortex in red, the calcium signal for neural activity, the cholinergic signal in secondary motor cortex in purple, and in black, the correlation between those two things. So here we see that there are moments when cholinergic release in V1 and M2 are highly correlated or are less correlated. Right? So the correlation in cholinergic release across space in the cortex is dynamic in time. And we can do that for the cholinergic signal, and we can do it also for the neural activity signal. And I'm going to show you the same sets of behavioral state changes. So here I'm going to show you the difference plots for the difference in correlations when the mouse goes from sitting to having high facial motion, so low to moderate arousal. And then I'm going to show you the difference plots for the difference between moderate arousal and high arousal. So on the left-hand side here is the difference in correlations across areas, but within color, so within channel, for low versus moderate arousal. So low facial motion activity to high facial motion activity. And here we see that on the left, there's not much change in the spatial correlation of cholinergic activity when the mouse goes from low to high facial motion energy, from low to moderate arousal, which is interesting because the amplitude of cholinergic signaling is extremely increased at this point, but the spatial correlations in that activity have not changed very much. Whereas in turn, the RCamp signal on the right-hand side here, even though there is sort of a modest and significant increase in neural activity, but also a very large increase in the correlations in neural activity across the cortex. The coolest part of this for me was this right-hand plot, which shows that, so red is an increase in correlations and blue is a decrease in correlations. And so here, when the mouse goes from having moderate arousal and high facial motion energy to very high arousal and running, even though we don't see a change in the amplitude of cholinergic signaling, we find that the cholinergic signals spatially decorrelate at that point. Right, so here you see basically a wholesale loss of correlation and cholinergic release across the cortex when the mouse starts to run. And we see very modest changes in the correlations in the neural activity at the same time point. So this tells us two things, right? One of them, again, is that the cholinergic signaling is not always a monolithic signal across the cortex. It is not always one signal. And depending on the behavioral state of the animal, you can see tighter correlations in the cholinergic signaling, or you can see all of that cholinergic signaling across the cortex decorrelate across areas and be very heterogeneous. This also tells us that the dynamics of both the neural activity and the cholinergic modulation can be quite distinct across these different types of behavioral state. Okay, so the last thing I'm going to throw at you here is something that is a little complicated, but maybe the most important aspect of all this. So everything I've shown you up to this point from this data set is we're talking about analysis of one signal by itself, the correlation of acetylcholine to acetylcholine, for instance, across areas. What we most want to know, though, is what is the relationship between the release of the neuromodulator and the local neuronal activity? Right, we would like to know, does the release of neuromodulatory signals like acetylcholine always have the same relationship with local neuronal firing? Or is it different across cortical areas, or is it different across behavioral states? So here, instead of showing you a measure of any independent signal, I'm showing you a plot of the correlation between the two signals. So this is a plot across the cortex of the correlation between acetylcholine release and local neuronal activity, right? So when the mouse is sitting quite still, low arousal and no facial motion, the correlations between the two signals are relatively modest. When the animal moves into this modest arousal state where the facial motion energy is high, but the mouse isn't yet running, we see a broad increase in the correlations between the two signals. However, when the mouse starts to run, we actually see some loss of the correlation between those two signals, but specifically in frontal cortex, not a global change, but specific to frontal cortical areas. Maybe easier to understand in these bottom plots, where here we're showing you the difference between behavioral states. So this is the difference in correlation between the signals when the animal changes between the behavioral states. So for instance, this is the high minus low face map and the locomotion minus high face map. So at each stage of arousal modification, right, we see a different set of changes in the correlations between acetylcholine and neuronal activity. So here, when the mouse goes from low facial motion activity to high facial motion activity, so very low arousal to modest arousal, we see a pretty broad increase in the correlations between acetylcholine and neural activity. However, when we take that additional step and the mouse starts to run, now we actually see a selective loss of the correlation between acetylcholine release and neural activity specific to frontal cortical areas, suggesting again that there's heterogeneity not just in the spatiotemporal dynamics of each of these sets of signals, but also in the coupling between acetylcholine and neuronal activity. Okay, so I'm going to run out of time, so I'm going to sum up here on this slide and then finish up. So again, just to reiterate, what we find is a really heterogeneous relationship between the release of the neuromodulator, release of acetylcholine, and the activity of populations of neurons in different cortical areas. We identified these three distinct behavioral states, and to be totally clear, right, we've identified three groupings of behavioral state that appear to be quite different at the level of neuromodulatory release and neuronal dynamics. They probably represent points along a spectrum, not fully divisible behavioral states, but rather 3 points along some arc of arousal. So what we find is that acetylcholine really goes up between low and moderate arousal, but doesn't increase in amplitude between moderate arousal and running, whereas the activity in cortical networks shows a fairly consistent increase across each of these levels. That gives us the results where the cholinergic release kind of peaks at this modest arousal level, but doesn't show much of a change, doesn't go up, actually goes down a little bit from there. And then when we look at the correlations, we find that cholinergic release is highly coordinated here, but decorrelated strongly upon locomotion. At the same time, local cortical circuits, as shown by our ECOG recordings and LFP recordings that I've shown you in different parts of this talk, local cortical circuits desynchronize upon arousal, right? When the mouse gets into that anticipatory mode, it's about to wake up and run, right? Local circuits desynchronize. But our large-scale cortical networks that I showed you in this data set, the large-scale cortical networks start to exhibit higher and higher correlations with increasing arousal. So here we have a divergence between the state-dependent changes in cortical activity at the local level, at small local microcircuits, versus the larger scale cortical networks that we think are important for behavior, like perception and cognition. Suggesting that the activity of the neuromodulatory influences on these circles may also be quite distinct when we're comparing the local cortical level to the sort of all over the cortex, the pan-cortical level, the cortical networks. Okay, So I'm going to skip this summary slide because I think I just said all of that, and instead point out that the work that I told you about today was bits and pieces of the work of 5 different postdoctoral fellows in my lab. So Quentin Perinaud did all of the behavioral analysis looking at state-dependent changes in perceptual performance. Katie Ferguson did all of the interneuron imaging data that I showed you at the very beginning. And Sweta Lohani, Andrew Moberly, and Habas Benisti worked together to do the imaging, the whole cortex imaging of acetylcholine release and local neuronal activity. And all of our imaging projects are done in close collaboration with Mike Higley's lab here at Yale, which has been an enormously productive collaboration. And of course, none of this is possible without support from the funding agencies. And thank you very much for listening. And if we still have lingering questions, we can stop and answer those.
Well, thank you, Jess. That was a very beautiful talk. Let me first just go through the chat questions. So Alexis Dagnino, he asked, is it possible to use virtual reality to carry out similar experiments? For example, a virtual water maze?
I don't know if we could make the mice think that they're swimming. But we certainly can make them think that they're navigating an environment. And in fact, one of the things we're doing literally right now is building a virtual environment for navigation to use with these wide field imaging techniques. So I can more fully answer your question about what the possibilities are in like 6 months. But yes, absolutely. It is totally possible.
Okay, and Alan Astudillo asks, have you analyzed causal dynamics interactions between areas apart from correlations? Yeah,
so this is a really interesting question, which I often touches a nerve when you talk to people who do things like fMRI for a living, right? Because historically, a lot of the analysis of data like this has been entirely about correlations, right? And the correlations are interesting and informative, but not the only thing you can look at. So we have also looked at this kind of data in 2 other ways. One is to use Granger causality types of analysis, where we are looking at the directionality of potentially causal interactions between areas, right? And that gives us a somewhat more clear view of patterns of potential flow, right, across areas. Somewhat more interesting, more recent, we've taken a graph theoretical approach to this data using a couple of different embedding techniques, actually. And that has turned out to be really, really interesting because that really allows us to look both at the sort of inward and outwardness of interactions between different areas and also at the patterns of co-activation ultimately. So we could go way down the path in talking about it, but it is certainly true that that's the obvious next step for dealing with this kind of data.
So your audio is off.
Oh, sorry. I have a question. With the data you show, it almost is kind of suggesting that alertness or wakefulness is more complicated. It's not just kind of low, moderate, and high. By looking at the differences when you start parcellating, where are more correlated and less correlated. For example, it seemed like the frontal area is more modulated by cholinergic signaling, I guess, when in the moderate, in what you call the moderate. And when the mice is moving, there's no change in terms of that, but the activity goes more caudal. So they're like kind of different. It's not just moderate and high, but like apples and oranges almost. You know, like they're doing something different in different regions.
Yeah, so absolutely. And one of the outcomes of a lot of the graph theory analysis that we did more recently is a collaborative endeavor, actually. Just went up on bioRxiv last week, where we took a broader approach to looking at behavioral state. And we actually find that if you take a sort of more holistic approach to this, what you find is that quiescence is clearly one state, right? The system returns to that low state over and over and over again. That the excursions, the departures into arousal are all over the place, right? It isn't that the whole brain goes from, oh, quiescent to moderate arousal to quiescent to moderate arousal. It's that it goes from quiescent to here, and then to here, and then to here, and then to here, right? And so, and you can separate those into groupings of states, but it's clearly the case the state is evolving on a faster time scale, right? And if you treat it that way, one of the things that you find is that there's potentially two general networks in the cortex that are modulated by these changes in behavioral state. There is a frontolateral network, and then there's this sort of medial posterior network. And interestingly, those two networks, the way that they fall out in space in the cortex, overlap with some of our like frontal cortex differences in cholinergic modulation, potentially. So you know, this is one way to deal with this data. When we take a hardcore quantitative approach to it, we find that underneath this level there is a much more complex and subtle level, which is a pain in the ass to deal with for analysis, right? but probably real.
Yeah, I imagine the next step is to look at this relationship with the cholinergic to the cell types, right?
Right, exactly, right. And also we're now doing this with neuropixels recording, so dense electrophysiology with the wide field imaging at the same time. And so we might get better temporal resolution of some of these changes that way. And also more headaches for data analysis.
Yeah, thank you. So Jocelyn, you have a question?
Hi, it's me again. Thanks for the talk. It was wonderful. I wanted to ask you first, there's a reason why do you, if I understand well, you put the electrode for stimulating the acetylcholine release in just one side of the brain? I mean, in one hemisphere? I understand that. Is that correct?
It's totally practical. There's not a conceptual reason. It's just that the implant itself covers up a chunk of cortex. And so we want at least one hemisphere totally clear for imaging. So if we implant bilaterally, we lose 65% of the imaging area.
Okay, so when you look at the maps of the whole brain, you just have like the one hemisphere like complete.
Yeah. So and we've, you know, we can see part of the other hemisphere when we have stimulating electrodes in, but we can't see the whole thing. So the stimulation electrode experiments were really a set of controls we did early on, and then we didn't implant stimulating electrodes after that because it made it very difficult to get interhemispheric data. Yeah. And we can implant the little ECOG electrodes much more easily, and those don't cover up nearly as much area. But for stimulation, they're huge. They're huge bipolar electrodes, right? They're like a telephone pole.
Okay, I got it. The other question, I think it was you talking about, and it was about the resolution. What is the resolution that you can get with this technique, with the last technique? And if you was thinking to, try it with different lines, like just see like the activity on one cell type, like deep interneurons, and in this case, I just thought for acetylcholine or that.
Yeah, no, that's a great point, and it's something we should totally do, is to look at the relationships of some of the interneurons that we know are sensitive to acetylcholine. So the scale here is about 50 microns. Okay. So one pixel ends up being about 50 microns. and you lose a little bit of we do a little spatial smoothing in this data. So you know, the point here is not to get cellular resolution, but rather to capture areas, right? But we have a whole separate set of things where we compare this with 2-photon imaging. So we can do a combined prep where we do 2-photon imaging of one area and whole cortex imaging using the 1-photon approach for the rest of the cortex. And that works beautifully. So you can get cellular resolution and whole cortex imaging at the same time. Yeah.
Okay, thanks.
Okay, so Will, you have a question?
Hi. If we have time, this could be a longer discussion if the time is running out. First, thank you for the presentation. Very elegant set of data. So I would like your view on an observation we have in human data. So in human experiments, we know that mild arousal improves performance along the data that you show, but extreme arousal hinders performance. So how do you see that in the context of your data?
That is a really cool question, right? So the performance in the mice actually does fall off with very, very high levels of arousal. And so you can see it, the very beginnings of that in the running mice. So when the mice run, the false alarm rate starts to increase. So their performance still gets better, but it's at a very specific cost, right? So they become more permissive, right? So they take more chances, right? And if you then do mean things like shock the mouse or give an intermittent noxious stimulus like an air puff to the face, you see that sort of accelerate that process, and the performance kind of falls apart, right? So our high arousal state is maybe just the tip of that. You know, you see just the beginning as the false alarm rates start to go up, they get a little more permissive in their responses. And then if you push them a little further, you know, performance kind of, you know, really falls apart. So the answer might be in part that our mice are very extensively habituated, right? So they are very, very comfortable in the setup, and running is anxiolytic for mice. So their stress levels go down when they run. So to see a truly hyper aroused state, like all the way to the far end of the range, we have to add something on top of that. I have a colleague who does consciousness experiments in humans where they look at the near threshold perceptual performance of their human subjects. And as you say, there are some clear correlates in those data for levels of arousal. In their data, they tend to throw out everything that's like hyper aroused because it isn't on point for their experiments. But the stuff that they throw out totally agrees with what you are pointing to.
It just comes to my next point, which is could be a matter of semantics, because I'm taking issue with the phrase that you used several times, that the neural population is modulated by the behavioral state. You repeated this several times. And what makes the behavioral state? I mean, it's the neural population that makes it. So something about the direction of the causality, which is the question of Alan. But if we know that increased arousal at some point drops the performance, then the sentence doesn't hold.
That is certainly true. There is evidence from some of our earlier work that you can decouple some of the things that I just described. That suggests at least the places that we have recorded, like primary visual cortex, does not appear to be a driver of the behavioral state, because in the sort of offset transitions and in some of our other work, you can decouple some of the elements. So you can decouple the neural changes from the behavioral state changes using other experimental manipulations. So at least for V1, the the neurons don't appear to be driving behavioral state change. I'm sure that if we looked more extensively throughout the brain, we would find other places like the brainstem, for instance, where that's not true, right? And it's, anytime you record neural activity, you're recording the combination of internally generated states with externally imposed stimulus states, right? So this is at its heart a chicken and egg problem, for sure.
Thank you.
Okay. I guess just before we let you go, I just have one last question, which is about the neuromodulators, right? So, and maybe it's kind of speculation that I'm asking. So how much you think looking at cholinergic signaling is going to reflect this, complex state, behavioral state question, and how much is going to be some kind of interaction between multiple neuromodulatory systems that, have some emerging thing that you don't see from one?
Chayu, if you were wearing green today, I'd call you a plant. Yes, absolutely. And in fact, I have a graduate student who, this week, has just started imaging mice in which we're looking at norepinephrine and acetylcholine. And also separate mice where we're looking at at norepinephrine and calcium activity, right? So it's a neural activity. So yes, our preliminary data suggests that the spatiotemporal dynamics and the coupling of norepinephrine to behavioral state is different than that of acetylcholine. So sometimes there are behavioral states where they appear to be very, very closely coupled, and behavioral states where they appear to have no relationship. So it's if the question is the brain simple or complex, the answer is always option B, right? So I'm sure that it's going to be complicated. We have another set of things ongoing right now where we're doing cholinergic imaging longitudinally during learning and performance of a task. And that data we haven't fully analyzed, but I think that may give us more insights as well. Here, we're just looking at spontaneous behavior. We may gain more insight from looking at more complex behaviors, I think.
Mm-hmm. Like tying maybe the second half of the talk with the first half.
Yeah, exactly. Yeah. So we'll get there.
Okay. I guess there are no more questions. Thank you so much. That was a beautiful talk.
Thank you for all those fantastic questions, and especially thank you for asking questions throughout, especially for Zoom talks. It's really, really nice to stop and have a conversation along the way. Yeah. So this was lots of fun.
Great. So we'll stay in touch. And thank you.
Thank you so much, everyone.
Thanks very much, Jess. Bye-bye.